Now I discuss about the nucleic acid. Nucleic acid are long chain macromolecules. Nucleic acid are long chain macromolecules which are formed by end-to-end polymerization of large number of repetitive unit called nucleotide. So nucleic acid is a organic acid. So nucleic acid is a organic acid which are present inside the nucleus of a cell, and it is made up of nucleotide. So nucleotide is the structural unit of nucleic acid. So nucleotide is the structural unit of nucleic acid. It is the structural unit of nucleotide is the structural unit of nucleic acid. So what are the nucleotides? Nucleotide are compound of carbon, hydrogen, oxygen, nitrogen and phosphorus. So the composition of nucleotide are carbon, hydrogen, oxygen, nitrogen and phosphorus. These are the composition of nucleotides. Okay. Then the structure of nucleotide. Of nucleotides. If in nucleotide there is three component, there is three component. These are the first a pentose sugar. Then the nitrogen bases, and third one the phosphate as phosphoric acid. For that we called nucleic acid. So the acid nature of nucleic acid is provided by the phosphoric acid or phosphate. Okay. So first component of nucleotide is pentose sugar. The pentose sugar of nucleic acid are grouped into ribose sugar and deoxyribose sugar. So what is the difference between ribose sugar and deoxyribose sugar? The structure of ribose sugar is like that. That is a furanose, first carbon, second carbon, third, fourth and fifth. So first carbon, first one, second one, third one, fourth one, and fifth one. Here the fifth group is CH2OH. Here H, OH, H, H, OH, H, OH, H, CH2OH. So that is the structure of a ribose sugar. If we observe the structure of deoxyribose sugar, then we observe that the second carbon in second carbon a oxygen molecule is absent, and oxygen molecule is absent. So the structure of deoxyribose sugar is like that. Here first carbon has H and OH group, second carbon has H and H only. Oxygen is absent there. For that, this ribose sugar is called deoxyribose sugar, means one oxygen molecule is absent in that structure. H, OH, H, CH2, OH. So if we observe the second carbon, an oxygen molecule is absent there. For that, this structure is called deoxyribose sugar, and that is the ribose sugar. Ribose sugar present in case of RNA. Ribose sugar is present in RNA. For that, the full form of RNA is ribonucleic acid, means it consists of ribose sugar, and this sugar is present in DNA. For that, the full form of DNA is deoxyribonucleic acid. Deoxyribonucleic acid means it composed of deoxyribose sugar. So the pentose sugar which are present in the nucleotide of a DNA, it is deoxyribose sugar. Okay. Then the next part is nitrogen bases. Nitrogen bases. We can also classify the nitrogen bases into two group. These are the purine. These are the purine and pyrimidine or pyrimidine. Purine and pyrimidine. In case of purine, there is two nitrogen bases, that is adenine and guanine. Adenine and guanine. Okay, A and G. These are the purine. And pyrimidine has three nitrogen bases, that these are the thymine. These are the thymine, T, cytosine, thymine, cytosine, C, and uracil. And uracil, thymine, cytosine, and uracil, U. These are the pyrimidine group. Then what are the difference between purine and pyrimidine bases? Purine has two rings. Purine has two rings. One hexagonal ring, and one pentagonal ring. And a nitrogen base is present, NH group is present in case of ninth place. And in case of pyrimidine is composed of only a hexagonal structure. This has a hexagonal structure. and the first place there a NH group is present. So first place a NH group is present. So that is the structural difference between the purine and pyrimidine. So here purine base is adenine and guanine and pyrimidine base is thymine, cytosine and uracil. Okay. Then the phosphate group. Phosphate group is H3PO4. Phosphate group means phosphoric acid. Phosphate present as phosphoric acid that is H3PO4, and the structure of phosphoric acid is like that: phosphate in center, 3 OH group, OH and double bond. That is the structure of a phosphoric acid. So these are the component of a nucleotide. These are the component of a nucleotide. So nucleotide is made up of, in case of RNA, they are ribose sugar, purine base adenine guanine, pyrimidine base cytosine and uracil in RNA. And in case of DNA, there is deoxyribose sugar, adenine, guanine, thymine and cytosine and a phosphoric acid. These are the component of the nucleic acid which are present in a DNA. Okay. So how nucleotide will form? At first, at first, here I draw the structure of a deoxyribonucleotide. Deoxyribonucleotide. So first the pentose sugar. First the pentose sugar is added. Is added or attached. Pentose sugar is attached with the with a nitrogen base. Pentose sugar is attached with a N2 base, and they formed nucleoside. So nucleoside is made up of pentose sugar and a nitrogen base. Then if nucleoside is attached with a phosphate with a phosphate or phosphoric acid H3PO4, then it formed the nucleotide. If the pentose sugar is ribose sugar, then it called ribonucleotide. If pentose sugar will deoxyribose sugar, then it called deoxyribonucleotide. Okay. So it is a pentose sugar. Okay. I draw here the deoxyribose sugar. It is the deoxyribose sugar. Then how nucleoside will form? If any nitrogen base attach at the first carbon of the pentose sugar. Here is the first carbon. Here is the first carbon. So the nitrogen bases are attached with the pentose sugar by its first carbon place. Okay. If here if here the purine base is attached, if here the purine base is attached, means adenine or guanine is attached with the pentose sugar, then it attached by its ninth place. If here pyrimidine is attached, means thymine, cytosine and uracil. If the pyrimidine is attached, then it attached by its first place. Okay. If purine is attached, then it attached by the ninth place. If pyrimidine is attached, then it attached by its first site. Okay. And here a bond will form. This bond is called glycosidic bond. This bond is called N-glycosidic N-glycosidic bond. So the bond is formed in between the pentose sugar and the nitrogen bases is N-glycosidic bond. Then it is a nucleoside. Then it is called the nucleoside. How nucleotide will form? If the H3PO4 means phosphate group is attached with the pentose sugar, then means nucleoside, then it is called nucleotide. So where does the phosphate gets attached? Where? In the pentose sugar, the phosphate can bind with two places. These two places are third carbon or fifth carbon. So the phosphate gets attached through third carbon of pentose sugar or the fifth carbon of pentose sugar Fine. In a nucleotide, only one phosphate group is present. If the phosphate get attached with the fifth carbon, then the 3 carbon place will free. So this end is called 3 prime. And if the phosphate get attached with the 3 carbon place, then fifth carbon place will free. Then the fifth carbon place will free. Then the fifth carbon place is called 5 prime. Okay, so that is the structure of a nucleotide. And the bond which is formed in between the pentose sugar and phosphate group is called ester bond. So in a nucleotide we can see that two bonds will form. One is N-glycosidic bond which is formed in between the nitrogen bases and pentose sugar, and an ester bond will formed in between the pentose sugar and phosphate group. Okay, so that is the structure of a nucleotide. Formation of nucleosides and nucleotide in case of RNA and DNA. I make a chart. Look it. First, the nitrogen base, N2 base. First the N2 base, then the sugar, name of nucleoside, then phosphate gets attached, and the nucleotide will form, Okay, so first the nitrogen base. So first the nitrogen base. First nitrogen base is adenine. Adenine can bind with the ribose sugar or deoxyribose sugar. If the adenine get attached with the ribose sugar, then the nucleoside is called adenosine. Then the nucleoside will called adenosine. Okay. Then phosphate get attached with adenosine. Then they formed adenylic acid or adenosine monophosphate. Adenosine monophosphate or AMP. Adenosine monophosphate. If the adenine attach with the deoxyribose, then it called deoxyadenosine. When the phosphate get attached with the deoxyadenosine, then it is called deoxyadenosine monophosphate or deoxyadenylic acid. If the adenine attached with the ribose sugar, means when adenine present in RNA, then they form the nucleoside called adenosine. When the adenosine attached with the phosphate, then it called adenosine monophosphate or adenylic acid. If the adenine gets attached with the deoxyribose, then the nucleoside is called deoxyadenosine. When the deoxyadenosine get attached with the phosphate group, then it called deoxyadenosine monophosphate or DAMP or deoxyadenylic acid. At same, if the guanine attach with the ribose, then it called guanosine. When it attach with the phosphate, it's called guanylic acid or guanosine monophosphate or GMP. If the guanine attach with the deoxyribose, then it called deoxyguanosine. The name of nucleoside is deoxyguanosine. When the deoxyguanosine is attached with the phosphate, then it's called deoxyguanylic acid or deoxyguanosine monophosphate or DGMP. That is the first adenine completed. Then the guanine. If guanine attach with combine with the ribose sugar, if guanine attach with the ribose sugar, then it is called guanosine. When it attach with the phosphate group, it's called GMP, guanosine monophosphate. If the guanine attach with the deoxyribose sugar, is attached with the deoxyribose sugar, then it is called deoxyguanosine. If deoxyguanosine attached with the phosphate, then it formed the DGMP. DGMP means deoxyguanosine monophosphate. Then the next nitrogen base is thymine. If thymine attached with the ribose sugar, then it called sorry, thymine is only present in DNA. For that, it combine with the deoxyribose sugar only, deoxyribose sugar only. Then it called deoxythymidine, then it called deoxythymidine. When it get attached with the phosphate, then it is called DTMP, means deoxythymidine monophosphate. Deoxythymidine monophosphate. Then the next group is uracil. Uracil is present only in RNA, so it binds with ribose sugar only. If the uracil get attached with the ribose sugar, then it is called uridine. Okay. If the uridine gets attached with the phosphate group, then it is called uridine monophosphate. Or UMP, uridine monophosphate. Then another and last base is cytosine. If cytosine can bind with ribose sugar and also with deoxyribose sugar, if it attach with the ribose sugar, then it called cytidine. If it attach with deoxyribose sugar, then it is called deoxycytidine. If they gets attached with the phosphate molecule, then cytidine is called CMP, means cytidine monophosphate. Then it is called cytidine monophosphate, CMP. If it binds with the deoxycytidine, then it is called deoxycytidine monophosphate. So these are the nucleotide. These are the nucleoside. These are the nucleotide present in DNA and RNA respectively. Okay. Next, I discuss about the nucleic acid. First, the classification of nucleic acid. So first, nucleic acid is classified into two groups. Nucleic acid are classified into two groups. These are the DNA and RNA. These are the DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Okay. Then we can also classify the RNA into two groups. These are the genetic RNA and non-genetic RNA. The RNA which act as the genetic material of any organism. The RNA which act as a genetic material of an organism is called genetic RNA. If in an organism where the RNA and DNA both are present, then DNA is the main genetic material. And then in organism where the DNA is the main genetic material, their RNA act as the non-genetic their RNA act as the non-genetic material. Non-genetic means they does not bear or does not transfer the genetic characteristics. They does not bear or heritates the genetic characteristic, but they helps in many biochemical reaction, okay, or physiological activities. Some non-genetic RNA also classified into many groups, such as the main three non-genetic RNA is rRNA, means ribosomal RNA, which are the structural unit of RNA. Then mRNA means messenger RNA, which transfer the message from the nucleus to the cytoplasm of a cell at the time of protein synthesis. Okay, M stand for messenger RNA, which transmit the message from nucleus to the cytoplasm of a cell at the time of protein synthesis. And then tRNA, T stand for transfer. Transfer RNA means which transfer or which transport the RNA, which the RNA which transport the amino acid from the cytoplasm to the place of protein synthesis, to the site of protein synthesis. These RNA called tRNA. So tRNA transfers the amino acid from cytoplasm, from one part of cytoplasm to the another part where the protein synthesis will occur. Okay. Another non-genetic RNA are snRNA, small nuclear RNA, which are present inside the nucleus. Small RNA which are present inside the nucleus are called snRNA. Another is hnRNA. HN means heterogeneous nuclear RNA, which are present inside the cytoplasm. hnRNA is the precursor of the mRNA. hnRNA is the precursor of the mRNA. Okay, so the hnRNA is the intermediate RNA which are formed during the mRNA synthesis from the DNA. Okay, so these are the nucleic acid in our body. You also remember that DNA is the stable genetic material. DNA is more stable than RNA. DNA is more stable than RNA because they lack the oxygen molecule in their pentose sugar or in their nucleotide. For that, DNA is more stable than RNA. For that, if any organism contains both the DNA and RNA, for higher stability, DNA act as their genetic material, and then RNA act as the non-genetic material. If any organism has RNA only, DNA will absent. At that case, RNA act as a genetic material. In some viruses, you all are known that RNA virus, ARNA virus is a TMV. So in TMV, the RNA act as the genetic material because the lack of DNA. If any organism contains both the DNA and RNA, then DNA is the main genetic material of that organism, and in that case, RNA act as the non-genetic material which performs many metabolism. Okay. Now I discuss about the DNA. DNA. The full form of DNA is deoxyribonucleic acid. So the DNA is composed of many deoxyribonucleotide. The the structural unit of DNA is deoxyribonucleotide. So the deoxyribonucleotide are of four types, as these are the DAMP, deoxy adenosine monophosphate, then DGMP, deoxy guanosine monophosphate, then DCMP, deoxy